Good afternoon. I want to thank you all for joining. I'm very excited to talk about how we executed extreme high throughput and efficient automated workflow to determine blood to plasma ratio. First, a little bit overview on blood to plasma ratio and its significance. This ratio measures the total amount of drug present in the blood and plasma. Blood to plasma ratio of greater than 1 is a consequence of drug distributing into red blood cells. Generally, pharmacokinetic parameters are usually determined by measuring drug concentration in plasma rather than whole blood. The parameters determined using plasma data could be misleading if drug distributes into the erythrocytes. In order to accurately measure the drug clearance, it is important to estimate blood to plasma ratio. Physiologically based pharmacokinetic modeling and simulation have been well established in pharma industry in the recent times. Blood to plasma ratio has been one of the important parameters along with other ADME and physicochemical properties of drug in predicting pharmacokinetic profile of the drugs. This table is from a review article on PBPK modeling and simulation in drug research and development by Zhuang et al. The table summarizes the data requirement for building a PBPK model in SimSip. SimSip is a modeling and simulation software. As we can see here, blood to plasma ratio is an important parameter in building the model for prediction of pharmacokinetic profile of the drugs. This classification system is from a review article by Miller et al. Emphasizing the importance of blood to plasma ratio value in PBPK modeling for first-in-human predictions using GastroPlus, which is another modeling and simulation software. Next, let's see the methods to determine blood to plasma ratio. The blood to plasma ratio is determined using in vitro methods rather than in vivo. There are different techniques established and each has its pros and cons. The blood to plasma ratio described here is adapted from a method by Yu et al. Test compound is spiked into fresh heparinized whole blood, reference red blood cells, and reference plasma. Following the incubation period, the whole blood is centrifuged. Both fractions of the whole blood, that is plasma and red blood cells, are analyzed by LCMS alongside reference samples. This is a tedious method as it takes time to separate both red blood cell and plasma fraction for reference samples as well as separation from whole blood post-incubation. It is also a labor-intensive procedure as incubation for whole blood is done in Eppendorf tubes to be able to get an aliquot of both plasma and RBC fractions. Post-incubation. Also, there are 3 freeze and thaw cycles to ensure cell lysis for better sample extraction. It takes about 5 to 6 hours to determine blood to plasma ratio for about 12 compounds using this method. This is a traditional method to determine the blood to plasma ratio where compound is incubated in whole blood at physiological pH and temperature, and post-incubation time an aliquot of whole blood is taken and rest of the whole blood is centrifuged for plasma extraction. Sample extraction technique is similar to the method described earlier. It has been observed that using this method, the blood to plasma ratio is underestimated, and this could be due to the sample extraction technique. And using this method, it takes about 4 to 5 hours to estimate blood to plasma ratio for 12 compounds. This method 3 is simplified and less tedious or labor intensive over previously described techniques. Here, the drug is measured in plasma, which is relatively easier to handle over blood. In this method, The control or reference plasma is separated from heparinized whole blood. Both control plasma and whole blood spiked with compound are incubated in parallel. Post-incubation, whole blood samples are centrifuged for plasma fractions. The compound in control plasma samples and plasma separated from whole blood samples post-incubation are measured by LC-MS analysis. The sample extraction technique is much simpler as it is only a direct protein precipitation method. This method takes about 3 to 4 hours to determine blood to plasma ratio for 12 compounds. Historically, the blood to plasma ratio methods have been low throughput. We have developed a high throughput method to determine blood to plasma ratio for 96 compounds at a given time using the method 3, which would have taken few days and labor to do the assay manually using any of the techniques described so far. This current slide shows overall workflow for high throughput screening to determine blood to plasma ratio. Firstly, control plasma is separated from heparinized whole blood. We use TCAN, which is a liquid handling robo system with a 96 channel pipetting head to add blood or plasma to 384 well plates. The orbital shaker on TCAN offers controlled shaking as well as heating options. The shaking prevents erythrocytes from settling down and heating maintains physiological temperature. Next, using TCAN, the compound is added to the blood and plasma containing 384 well plates. The plates are incubated for 60 minutes. Post-incubation, the blood plate is centrifuged to separate plasma. The Agilent Bravo platform with 384 well pipette head is used to transfer the plasma fraction from centrifuged whole blood samples. Also using Bravo, acetonitrile containing internal standard and aliquots of plasma fraction are added to 384 well plates. The supernatant from centrifuged 384 well plates is collected using Bravo for final LC-MS analysis. Let's see the brief protocol using TCAN and Bravo liquid handling robo systems. 147 microliter of blood or reference plasma sample is pipetted 4 times into 384 deep well plates. 3 microliter of test compounds is added to the blood or reference plasma in quadruplicates. Plates are incubated for 1 hour at 37 degrees Celsius. Using Bravo, acetonitrile containing internal standard is added to 384 well plates. Aliquot of plasma supernatant from centrifuged blood samples and aliquot from reference plasma samples are added to 384 well plates containing pre-added acetonitrile with internal standard. 384 deep well plate makes it easy to collect plasma fraction from whole blood samples post-incubation. Discovery Quant software from AB SciEx is an automated MRM tuning and optimization for high-throughput drug analysis. It reduces significant amount of time that would take to manually tune the compounds. Compounds are tuned at a rate of 60 seconds per compound. Q1, Q3 scan, declustering potential collagen energy parameters are stored in database. The supernatant collected using Bravo from centrifuged plasma fraction with acetonitrile containing internal standard are analyzed using our generic conditions on C18 column. These are the results from the experiments done on different days to determine interassay variability. The results from interassay variability experiments are outlined in the table below. The following compounds were selected for investigation based on previous literature as well as in-house data generated from manual assay. The percentage variability for all the compounds between 5 validation sets is lesser than 20% except for chlorthalidone. The higher variability in chlorthalidone has been observed in manual assay as well. The results in the table is to show intra-assay variability. The data is from one of the validation sets. Intra-assay variability between the replicates was low except in case of methazolamide where wider variability occurs due to quantification of very small residual amounts of compound in plasma. Based on the results, acceptable percent CV were obtained to assess inter and intra-assay variability. The graph illustrates the inter-assay reproducibility of the blood to plasma ratio values for the listed compounds. Verapamil, diltiazem, midazolam are considered to be negative controls or low partitioning compounds, whereas chlorthalidone and methazolamide are considered positive controls. Chlorthalidone was found to be a poor positive control owing to its low observed blood to plasma ratio and moderate variability, which was also observed in manual assay. Methazolamide was found to be a better positive control. Good agreement is observed in the graph illustrated between the blood to plasma ratio values obtained from high throughput screening versus manual method. Here, I want to emphasize the importance of blood to plasma ratio value and how the value can differ between the compounds. High throughput method developed to determine blood to plasma ratio enables to screen large number of compounds in early drug development process. High throughput screening technique developed to determine blood to plasma ratio utilizes the automated liquid handling robotic platforms for rapid testing of large number of compounds. The current automated method eliminates labor-intensive steps with faster turnaround time and quality data. High-throughput screening method to determine blood to plasma ratio saves significant time in testing large number of compounds and generating data at a quicker pace in comparison to manual method. It takes about 4 hours to test 12 compounds manually, and also it is tedious and laborious to collect plasma fractions from whole blood samples manually post-incubation. Whereas 96 compounds at a given time can be done using high throughput method under 4 hours utilizing liquid handling systems. The experiment can be handled by one scientist in high throughput screening method. Also, high throughput screening method utilizes minimal labware in comparison to the manual method. In the manual method, it is challenging to extract low volumes of plasma from 96 well plate from centrifuge whole blood post-incubation. The well in 96 well plate is much wider in comparison to the 384 deep well plate, and this makes it difficult to extract lower volumes of plasma, or higher volumes of blood has to be used in 96 well plate during incubation, which would be lot more reagents and material cost. The take-home message here would be high throughput screening in 384 well format to determine blood to plasma ratio is done at a quicker pace with more number of samples obtaining high quality data. Robust high throughput screening method has been developed to measure blood to plasma ratio, and here are the conclusions for the developed method. A state-of-the-art high-throughput blood-to-plasma partitioning assay in 384 well format was developed and implemented. The method enables running 96 compounds in quadruplicates at a time in 384 well plate. A wide range of robotics like TCAN and Bravo have been used to perform the assay. The technique developed generates quality data at faster pace using less materials. The automated method developed to determine blood to plasma ratio can be used to routinely screen large number of compounds efficiently during early drug development phases. The success of developing and implementing this high-throughput blood-to-plasma ratio assay is an example of powerful team effort within Cyprotex. And with this, I would like to thank entire team of Cyprotex and everyone listening to the talk virtually.